Now, can you please? Yeah. Hi, everyone. Thank you for joining this talk today. So today, I'll be moderating this talk. My name is Maimouna Al-Musawa. I'm a PhD student at CTERM UKM Malaysia. I'll be moderating this talk along with my colleague, Carlos. He is also a PhD student from University of Coimbra, Portugal. So today we have a special guest, Dr. Ana Claudia from from the Federal University of Sao Paulo, Brazil. She's going to talk about the extracellular vesicles and Chagas disease. We're very excited for this talk. So thank you so much for accepting our invitation. And let me introduce Dr. Ana Claudia. Dr. Ana Claudia did her PhD in biological sciences in the graduate program Biology of Parasite-Host Relationship from the University of Sao Paulo in 2004. She completed her postdoctoral studies at the Institute of Chemistry of University of Sao Paulo from the years 2004 to 2008. And at the University of Georgia in the United States from the years 2010 to 2011. Currently, she's an associate professor at Federal University of Sao Paulo, Brazil, with experience in parasitic protozoology, focusing on the characterization of EVs isolated from the parasite and the mechanisms of interaction with the host. She's also noted for her contribution as a full member of the board and the scientific committee of the International Society for Extracellular Vesicles, ICEV, from the year 2016 to 2020. And she's also an associate editor of the Journal of Extracellular Vesicles, and also Journal of Extracellular Cellular and Infection Microbiology, and the Brazilian Journal of Health Research. She's currently the communication coordinator of the ICEV Society for the period of 2022 to 2024, and the network of researchers in extracellular vesicles in Latin America, Latin EV, since 2016. So Dr. Anna, thank you so much for accepting our invitation, and you may start your presentation and your talk. And just a reminder to our attendees that you can just place your questions in the chat box. We will attend to your questions after the talk. Thank you so much.
Can you hear me? Can you hear me? Can you see my presentation? And good afternoon, good night, and good morning for all colleagues. And a pleasure to support SNEV, and a pleasure to my second talk here in this amazing program to development to invite people to talk about science. My talk is extracellular vesicles and Chagas disease is a big problem in Latin America. This is a timeline, this first detection of Chagas disease, but it's the old parasite. These isolating mummies in Peru and Chile in the South America, and the first evidence, these infections in Latin America for this girl, her name is Berenice, and after that, Dr. Carlos Chagas, because the name of this disease is because this doctor is a Brazilian doctor. He's isolated the parasite from these girls and started to develop the treatment, and after that, he showed the life cycle of the parasite. You move to another place, another countries to found this Chagas disease in Latin America is a big problem, but this is Dr. Chagas, Carlos Chagas, here with Dr. Einstein. He's a Berenice, this is all the pictures about that, is the first place to isolate parasite in Brazil in 1999. And this is these infections transmit for the vector. This one the species is Trypanosoma infestans. 8 million people affect in this Chagas disease, a chronic phase. 10,000 people die to complication of the Chagas diseases. It's just available 2 drugs to treat. Sometimes the people discover the disease is the chronic phase, but a big problem. But WHO consider the neglected diseases, but no or little interest from the big pharmaceutical companies to develop a new treatment for the Chagas diseases. But we have a big problem around the road because the people from endemic area in Central America and South America move for another countries in Europe and as well in Japan or the Australia. And these people is a chronic phase, and the blood bank does not detect the exam for detect the antibody against the two parasite, and they spread a lot of infection to Chagas diseases in Europe. In Latin America, we control in the blood banks, but in US, you have a lot of problems now because this vector, when the species migration for the Texas, New Mexico, and California too, we found this transmission for the vesicles now. Now is a big problem. The transmission is 8% is vectorial, but in the south of Brazil and the southeast, we control completely the vector transmission. Now in the north of Brazil, the açaí fruit is the oral infections, 10%. The mother to fetus, blood banks, you have this not in Brazil, but the rest of the world is a big problem. Organ transplant, and sometimes in vivo model, you can transmit the Chagas disease. But we have the natural course. For example, in the feces of the Trypanosoma, the vector transmit the bite people and transmitting the feces of the parasite. We have the acute phase and chronic phase. The acute phase, you have a lot of parasitemia and they start a production of the humoral immune response. And after months, 4 or 8 weeks to move to the chronic phase, and some people used asymptomatic, some people 40% symptomatic forms to cardiopathy, mega esophageal and colon and cardiodigestive forms. The parasite is still in the organs and proliferation that. But if the efficient to treat this phase, acute phase, you control these diseases. Our goal to understand what happened this parasite in the acute and chronic phase to move the chronic phase. But the Trypanosoma cruzi, extracellular vesicles, how they involve the parasite. For example, this is, as you can see here, the tripomastigotes forms, the forms to infect the human and contain the plasma membrane glycoconjugates to induce and host immune systems from the cellular and the humoral immune systems. And the lytic antibody against glycoconjugates start this acute phase, but you have a lot of cells to participate in these events to promote these diseases. But you have a lot of questions. How the tripomastigotes release vesicles and contain these glycoconjugates like mucins, and these contain these EVs are soluble. These mucins interact directly to the plasma membranes to contain EVs and contain some receptors in macrophage or monocyte or dendritic cells and induce the cascade to MAP kinase and induce the synthesis of cytokines and chemokines. You have a lot of questions, but we need to move to understand when you start the studies of EVs. In the place when I finished the excellent lab to Dr. Walter Colli, is this my ex-supervisor, my first postdoc, starting 1979 with this Dr. José Franco da Silveira, is the first person in Brazil to isolate EVs from the epimastigotes forms from development in the vector. And these forms promote the proliferation of this parasite in the vector. Dr. Frank showed the first time the parasite release vesicles. The next paper about this in the Dr. Marinei Gonçalves, she showed the tripomastigotes forms shedding EVs, and this Dr. Walter Colli and Dr. Maria Júlia, my ex-supervisor, my first postdoc, showed in 1991. The second article about this parasite, the Trypanosoma cruzi, this protozoa parasite, release EVs. But how to isolation characterization disease. The people is 20 years ago don't believe the parasite release disease, but we start the group from Spain, Canada, and me in Brazil to develop the guideline to promote these interactions and how to isolation, characterization, how to approach to use. And we organized the guideline we published last year. This is, as you can visit in the journal Extracellular Biology, to purification and characterization extracellular vesicles from the whole parasite, protozoa and helminths parasite, and how to use in the functional assays. And this is, according to this ICEV, is very important to guide the people to start to work to parasite. And this is how to our approach. As in the first image, as you can see, the scan electronic microscopy, this is epithelial cells here. As you can see, these triple mastigotes form in red, and the triple mastigotes release vesicles. And what happened these vesicles and what happened these interactions, okay? But the first you needed to isolate in these, we collect in vitro or in vivo because you maintain the in vivo animals infected with the triple mastigotes forms. And we collect ultracentrifusion after that, or use the centrifugations, not use if you depends your approach, but use the sac column. And as you can see here, we have antibody against the glycoconjugates. This fractions we collect and put in the pool, and you would detect the smear is like mucins and TCH5. This glycoconjugate expressed in plasma membrane from the parasite contain these EVs isolate from the parasite. And as you can see more pictures about this parasite, here is like electronic microscopy, scan electronic microscopy. This is a plasma membrane from the parasite, and the parasite spontaneously release these EVs, and the 99% the parasite is still alive, is don't die, is maintain this viability very good. And we have more electronic microscopy. As you can see here, these vesicles is the fragilar pocket release vesicles in the plasma membrane from the parasite too. We published a long time ago these pictures, and this is more example for these forms to still in the human infection, sorry. As you can see here, the triple mastigote release EVs2, okay? But we show the parasite release vesicles. The another question, how the composition these EVs isolate from the parasite? Well, this is a plasma membrane from the triple mastigotes forms, the forms to infect a human, and contain glycoconjugates like transialidase, GP85, it is a superfamily, the glycoconjugate we express in the parasite, and alpha-galactoses. These EVs contain the same markers, the same glycoconjugates. From this, we have the first questions. We contain this smear is from mucins and TCH5, I showed before, and this proteomic analysis, we found RNA, we found MASP from this virulence factor, we found mucins, TCH5, and transialidase. These EVs show 6% of the protein correspond to the glycoconjugates from the plasma membrane from the parasite. And we show, we have the second question, how many populations the triple mastigote release EVs? We use this FFF. This is a fractionate EVs because we have this principle as a hydrodynamic size, and we channel flows, cross flows, and separation nanoparticles. And as you can see here, we found two different populations, the parasite release. One is small than the name EV1, and second EV2. And after that, after that, after that, as you can see in this microscopy, we have the two different population of EVs. And this is a proteomic analysis from the EV1 and EV2. As you can see, transialidase, we don't detect a lot mucins, but we detect the ELISA. We detect the peptidases, heat show proteins, and a lot of proteosomes in the another type of EVs. But we propose, we have the heterogeneous EVs compositions and subpopulation, but we have the group of the small EVs, like the 50 and 100 nanometers, and another type of group 2 EVs, 100 to 250 nanometers, okay? But we have a second question. The biogenesis of extracellular vesicles released from the parasite. You use this scorch machinery, and with development, you can see the scorch components in Trypanosomatids. We found the Trypanosoma brucei, Leishmania major, or autotype for the species of Leishmania, and Trypanosoma cruzi, and the odd type of this protozoa parasite. And this, we started to development EVs to found the biogenesis of EVs, our project. And we optimization the CRISPR-Cas9 system for genome editing. And we have to knock out parasite to escort machinery, VPS escorting machinery. And as you can see, this parasite, this knockout, you development and you clone 2 knockouts and decrease the number of particles and in the 2 different forms of the parasite. And you have more, the VPS knockout reduce the infectivity in the host cells and decrease the number of infected cells in the knockout than the wild type parasite, and decrease the number of the parasite infections. And this escort machineries is like, I don't know, like the exosomes, but these EVs isolate from the parasite, reduce the infection in the human cells. And how about the functional assays? We labeling these with the TO orange and the incubation to human monocyte, and they use this approach during the time, 10 minutes, 1 hour. And as you can see, the uptake from the human monocytes, and they use the image stream, 7% of the cells incubation of EVs in 10 minutes we detect the EVs in the cytoplasm of the host cells. As you can see, the control, you find some not EVs, but it's very important because it's very quickly the interaction is EVs. It is uptake for the HeLa cells in 1 hour, as you can see these EVs in the cytoplasm from the host cells. It's very quick interaction. These EVs isolate from the parasite in the epithelial cells, monocyte cells, and we have very interacts another sub-project for our grant. One with incubation EVs from the human monocyte, we found this after 24 hours, we found this monocyte is completely exchanged this morphology. Our question, these EVs differentiate monocyte and macrophage or not? This our goal now we, as you can see here, we found this completely different morphology, this monocyte after 24 hours to incubation to EVs isolate from the parasite. Our goal now to understand what happened in these interactions if we move to morphology and the function to macrophage M1 or M2. This is a confocal macroscopy. As you can see, the same result after 24 hours, completely exchange this morphology, and we start to work these functions. And these EVs want incubation to monocyte to translocation NF-kappa B. This NF-kappa B contain endogenous GFP, as you can see here, a lot of spots to green fluorescence. And we use this, as you can see here, and these EVs released from the parasite. These EVs contain different population of the parasite and reduce the infection in the human cells. These interactions is very important because in the monocyte, in 10 minutes, you found these EVs in the cytoplasm of the cells and translocation the NF-kappa B to the nuclei and induce the cytokines. No, I show next. But what kind of receptor or pathway to involve? Because the several colleagues who work the parasite show the mucins interacted to toll-like receptor 2. And they use this macrophage isolate from knockout mice for toll-like 2 and the wild type. And after that, collect this macrophage and the incubation to EVs to different concentrations. After that, my group show this production, the cytokines and nitric oxides. As you can see, the second, this experiment, this macrophage isolate from the knockout mice, reduce these cytokine productions. And we published in 2025 in June Extracellular Vesicles. This is the first time these EVs isolate from the parasite, interact like receptor 2, and depends this to induce the innate immune response. And what about the parasite invasions? The parasite invasions is contact to plasma membrane from the host cells in the acute phase and maintain these infections. And the parasite is still proliferation, the mastigotes forms in the plasma membrane from the parasite. And that approach this mechanism this invasion from these forms to develop the human state, our questions, if we treated these host cells with EVs, and after that, incubation to triple mastigotes, it's possible to increase this invasion of the host cells. And as you can see here, this is an image to show these host cells, epithelial cells, because it's very important, the first contact in the epithelial cells. As you can see this, a lot of mastigotes, this intracellular parasite, when a pre-treated these culture cells with EVs isolate from the parasite, increase the number of infected cells, then control, don't add EVs. These EVs induce more invasions and induce the more infected cells and induce more parasite proliferation. And as you can see here, our next question is the toll-like receptor, if you incubation with EVs induce more infections, yes. We have these C8O cells to express toll-like receptor, and you pre-treated with EVs isolate from the parasite. After that, infections, and 24 hours after, we count the number of infected cells. When isolate the parasite, you use those different concentration, the parasite per ml in this assay. We increase the number of the parasite in the infected cells. And we move to in vivo assays. We label EVs from isolate from the parasite. After that, inject the animal model. And I use two different types of these strains. One strain virulent and the other non-virulent, but the same strains, but we pass 20 times the parasite in the animal model, bulbous mice. As you can see here, after 24 hours, we fix these organs, and you show, as you can see here, the intensity of the fluorescence, the EVs. EVs move for other organs, spleen, heart, brain, lung, intestine, and liver, of course. But as you can see here, in 24 hours, we found a lot of EVs in many organs. These EVs prepare the microenvironment to parasite invasion and proliferation and move to chronic phase. Our goal to understand what happened is. And we work in the several years in the animal model. We pre-treat the animal with the EVs, after that infections, and I look at the parasitemia and mortality, and in 15 days, we collect some organs to count the numbers of the mastigoat's nests, the intracellular parasite in tissue, and this look this, and this first result is not statistically significant, but increase the number of parasite in blood for the animal model, but the animals die before the control. And the very interesting, this is a heart tissue. As you can see, when the animal pre-treated with this, we found more a mastigoat's nest, more infection in the tissue than the control. These EVs induce more invasion in the tissue. But and finally, our suggest the mechanism, the tripomastigotes form release two types of groups of EVs, one small and other bigger than this EV1. This group of EV1 and EV2 and contain the glycoconjugate isolate from the plasma membrane from the parasite. And the interaction depends the toll-like receptor. We have more data about the MAP kinase induce and the activation, and induce the proliferation, the translocation NF-kappa B to the nuclei, and induce the pro-inflammatory response and IPTENs. Why this induce the pro-inflammatories and no control the diseases? Because it spread more cells. The cells come to this microenvironment, and these EVs release from the parasite or macrophage and contain some glycoconjugates from the parasite, increase invasion. We have more data about the insect factor because I don't have time to talk today, but this triatom, the vector, we feed to EVs isolates from the form to development here, and this increase the number of this parasite, and the vector show is the interactions is I have some considerations about this parasite in the intestine from the vector interactions. And it's a pleasure to finish my talk here. I don't work alone because my students is so nice and they have a lot of support. Dr. Patricia Schander, from my colleague, the same university, and you share the same lab. Dr. Sergio Schenkman to help me to the development, the CRISPR-Cas9 from the use to VPS from the parasite. Oops. Sorry, and this is Rodrigo Soares and Paula Nogueira from Fiocruz. We have a collaboration to Nectar Helgev Hudings from the Weizmann Institute of Israel, and my ex-supervisor, Dr. Igor Correa de Almeida from the UTEP from US, and Dr. Carmen Bezerra Fernandes from the Barcelona. And I need to thank for the agents to support my research for more than 22 years in Brazil, and the CAP is a federal institute to support FAPESP, our agency in the state, and I live in São Paulo and CNPq. And I would like to share my group, my excellent students, my collaboration, Dr. Blima Fuchs, and thanks so much for invitation. Thanks so much for the opportunity to talk in my research.
So thank you so much, Dr. Ana Claudia Torrecilhas. It was a really nice talk, and I just want to add that it was inspiring to see how, I mean, people went from not believing that EVs from this kind of parasites can have an impact to come to a guideline published in Journal of Extracellular Biology. So thank you for sharing that. It's really amazing and inspiring to see this. And I think it's also really nice to show this to our colleagues. I'm just seeing the chat box. We have here a question. I might have a question for you later on, but let's start with the participants. I don't know the name. It's someone that is called user. I believe it might not be the real name. If you want to unmute yourself and ask your question, please do it. Otherwise, I can do it for you. Are you here? Okay, so maybe I can ask the question. So the person was asking if, in the case of labeling the exosomes for the fluorescence labeling of the extracellular vesicles, if you to see it on the microscope, if you need to do once again like a re-isolation of the extracellular vesicles after doing the labeling, for instance, using the exo kit.
I don't use the exo kit. I use the SEC column in-house because part of my postdoc, the people in 979 use the SEC column to isolate these EVs from the parasite. We collect the supernatant of these infected cells. After that, centrifugation to pellet the parasite. And incubation for another media, we wash 5 times. Incubation for the media without FBS. We add the glucose, 5%, add more. And after 2 hours, we collect the supernatant and check the viability of the parasite. After to use the SAC column and follow the fractions positive to glycoconjugates and the pool used to check the NTA, the concentration and the size. And after that, use this to label it at 1 hour, 37 degrees, depends the protocol. And after that, you ultracentrifuge 2 times. And after that, you check the NTA and check the fluorescence and move to functional assays. This is a very quick approach. Is a lot of steps, but I prefer to use the commercial kit because we established this protocol more than 25 years in my lab.
I see. Just a quick follow-up on that. For your in vivo studies, the biodistribution, which kind of dye did you use?
Deer.
Okay. Thank you.
Okay, but if any, please contact me. If any people have any protocol to in vivo or in vitro assays, I'm so open to send this protocol. It's very easy. It's not difficult, but you need to isolate 10 to 9 or 10 to 10 particles per each animal. It's a lot.
Then we have another question. Arefe Cardani, do you want to unmute yourself and ask the question?
Do you hear me?
Yes.
Yes.
Okay, thanks a lot for the great talk. I had a question regarding the uptake route of this EVs into the cells that you tested the EVs on. You mentioned that the EVs are working through TLR2 and they activate the NF-kappa B pathway through TLR2. So I was wondering if you also checked the internalization routes of these EVs in your targeted cells, and if you have any idea if this TLR2 is also responsible for the internalization and the activation.
Yes, the toll-like receptor 2 is because the parasite infect the whole cells, but it's not. I don't know how many % of the parasite depend toll-like receptor 2 to invasion or the dimer toll-like receptor 2 and 6 to infect invasion whole cells. I don't know because the parasite should interact in 20 minutes. The parasite is moved to the cytoplasm, is a vacuoparasitophore, after that escape and proliferation in the cytoplasm. But I don't know the % of the parasite, depends the toll-like receptor to invasion, but the toll-like receptor interact the pumps from mucins is one the type of the pump to interact toll-like receptor 2 and 6 and induce a lot of pro-inflammatory response. But when the uptake depends from some receptors, but this directly uptake in the whole cells is a move to the periphery of the nuclei of the whole cells. Our project moved to understand if what kind the RNA in these EVs from the parasite or some lipids or proteins modulate, and we moved to study this what happened in the nuclei from the whole cells. is this question, is control the transduction for the host cells. What happened? Because when the incubation of EVs from the parasite with these cells induce the pro-inflammatory response in 24 hours, after 72 hours, completely exchange from this cytokine production. But I need to understand this, what happened. But I don't know how many percent the toll-like receptors maintain, and maybe one the mechanism, this depends the toll-like receptor, but the parasite is very quickly to invasion a lot of host cells, endothelial or epithelial cells.
Thank you. Thank you very much.
Okay.
Thank you. So we have more questions in the chat box. Maimona, would you like to ask the question?
Yeah. Okay. Yeah. Thank you so much for the interesting talk. And I'm very amazed by your journey, and all the research that you've been doing. So you have a quite established protocol to begin with, right? So this question came to my mind. If somebody wants to study EVs from any kind of parasite that has not been studied before, different countries or different regions have different pandemic with parasites, right? So what's your advice, how to start with the research? I mean, with the isolation method, what to look at, and learning from your journey and from your experience?
It's so hard to start, but I needed to, the first, how the question, because we have a lot of questions to understand what happened this parasite in the host. How the parasite maintain for many, many years in the host. Now we'll have some light what happened, this mechanism to maintain the parasite, and we need to select different strains of the parasite. For example, Trypanosoma cruzi, we found 6 subtypes of strains, the family. You need to select some very virulence, no virulence. The first, we need to select this approach to understand, because there's modulation, different immune system, host immune system. Second, you need to antibody against some proteins or glycoproteins or lipids. We don't find the commercial to buy. With development, you produce the monoclonal antibody against this, because it's very easy to follow these EVs. And you have some machinery to test, because we work to particles to check to NTA or cytoflex to see how many particles you need to use to understand these functional assays. Is very important. But the first, you need to understand these infections. You need to understand how the parasite interactions. You need to select several strains to understand, because some strains induce low immune response, but some strains high level of the cytokines and increase a lot of infections. The first, and move to characterization and move to functional assays. But my suggestion, you need to perform some experiment to produce polyclonal or monoclonal antibody against some glycoconjugates from the plasma membrane from the parasite. This is so good to move to understand this characterization and the functional assays.
Yeah, amazing. Thank you so much. Yeah, thanks for the answer.
Welcome.
Thank you, Maimona. So we have another question from Amena. Will you like to unmute and ask the question directly?
Yes. Hello. Thank you, Dr. Anna, for this great talk. I have a question. My question is the cell who, like host cell who infected by the parasite, by the stress, by the way of infection, those cells also, did you check that they are increase or release more EVs, and that EVs can be important also to like immune response to the thing that you follow by the parasite EVs? I mean, for that cell also, did you verify this, or because we know that cells, when the stress situation or something like an infection, they can release EVs also. What is the difference is EVs from the cells itself or parasite?
Okay, this is a very challenging. We published in 2020 article, my PhD student, Andrea Cronenberg, we show when infected macrophage, human macrophage, and after our 24 hour, we collect the supernatant. These EVs contain some glycoconjugates because we performed the proteomic analysis. You found the mucins and transalidase in these EVs. These EVs from the macrophage infected with the Trypanosoma cruzi, tripomasto forms. But we performed the experiment, we found this. These EVs from the host cells contain some markers from the parasite. And we moved to understand what happened in the chronic phase, and we collected a lot samples from the chronic patients because you have the public hospital here in São Paulo and the Institute of Heart from University of São Paulo. The people helped me to collect the chronic patient with Chagas disease because it's very difficult to find the acute phase here in the south of Brazil. We found the acute phase in the north because the people eat açaí or some juice and contain the forms to transmit the disease. And we isolate these EVs from the chronic patients, and they perform some experiment, these EVs, to see if found some markers, biomarkers from the parasite. We found maybe these EVs release from the cells. I don't know if the parasite, because the parasite in chronic phase is still in the organs. But if the cells infected contain these markers, and maybe we should follow these patients during the treatment. But they decrease these EVs. Our goal now to understand this. If you decrease the number of EVs, maybe is the treatment is okay, use the benzoidazole or nifurtimox, the drugs to decrease the number of the parasite. But we found the human macrophage, when in fact these EVs isolate this macrophage, contains some markers from the parasite.
Yeah, thank you so much.
Welcome.
Thank you, Amanda, for the question. So I don't see any other question in the chat box, but I have a question. I really like your results about giving EVs to the animals and then seeing, and then you showed that the accumulation of the parasite in the organs is different when, I mean, the EVs somehow change the microenvironment of the organs. So my question, it seems to me that is something, somehow the EVs are changing the ECM, the extracellular matrix of the organs. Do you know, do you have any idea? Are you planning to look into that to understand why are the parasites targeting better to the organs after the EVs? Somehow they are changing something. So I'm just curious if you have anything about it or if you are planning to look into that.
Yes, excellent question, Carlos. My PhD student, her name is Mariana Ottomano. She's started to, we work through the bioprinting to the cardiomyocytes to the organoids. We start to now standardization this to understand what happened, because the heart and the intestine, our goal to understand this disease, what happened. In my view, but we standardization because it's not easy to work the organoids and the bioprinting to cells. But I would like to understand, it's very quickly. We found the disease in 24 hours. The one I published in 2009, the paper in vivo model to EVs, the people don't believe these EVs modulate the microenvironment in the heart. But in my view now, because you have more, we have this biodistribution in vivo now. We have some machinery like the IVs, the image stream to see the interaction, the uptake. In my view, these EVs, the monocyte move to chemotaxi for the chemokines because the EVs induce this in the heart or intestine. Monocyte move to the organs, and the EVs isolate from the parasite or infected cells and contain these glycoconjugates, help to to move the monocyte maybe in macrophage and still there and modulate the microenvironment. This is my question about is possible this. For
like
this,
a
we
immune
work this.
infiltration.
Yes, yes. And
I
in
see.
my, after a lot of my PhD at now, our goal to understand what happened, how the parasite is still alive during the third years or 4 years for the chronic phase from the Chagas disease. Maybe the chronic phase, the people don't die from the Chagas disease, die to the another type of the disease, but some people to asymptomatic die because the intestine or the heart has a lot of problems, but the parasite is still there. But how the immune system is not die, is not control these infections? Because in my view, the parasite modulate this microenvironment. One the reason we start to work in the biodistribution in vivo, and after that, we understand each type of organs. We start to bioprinting and the organize in the heart, and you move to intestine and maybe the brain. It's very quickly, 24 hours, you detect. Is a lot of challenge, Carlos. Is maybe it's more 6 or 10 years to work in this project. It's not easy, but
I
so
was
excited.
curious because I think it's I mean, it can be challenging to find the molecule, especially in the organ, because I mean it's a lot of molecules. So I was wondering if you were planning about that and how to get that, but it seems that you are already planning that.
Yes,
And that's
one
great.
that
Thank you for your answer.
yes, yes, and thank you for your question because it's a challenge to understand the disease. Why the parasite, like leishmania or malaria, the plasmodium, why the parasite is still there? And the immune system is not control this infection disease, don't
Especially
control the
because
parasite proliferation.
especially because you show that after incubating the EVs with the monocytes, then they become macrophages or they start the migration. So it seems that there is a response from the immune system, but somehow they can stay for long in the organs. So it's quite tricky or weird because it doesn't make sense to activate the immune system, but then stay there forever or in the chronic sense.
Yes, but how the challenge this is now, I have another PhD student, is Juliana. She worked in this project to monocyte and macrophage. But when incubation, this PBMC, we found different markers in the cells. I need to, we move it to understand if these cells, one, the morphology, I don't know yet this macrophage, move it to M1 or M2 macrophage.
Yeah.
This is our goal for the another project to understand this.
I see. Well, thank you for your answer. I think it was quite clear. Well, I don't see any other questions, so I think we can close the session. And thank you once again for coming, for presenting these interesting results, and thank you everyone for joining and asking your questions. Hope to see you in the next SNEV event. Thank
Of
you and
course,
have a nice day.
my message for young scientists, please still in this field, because the ISAF, this EVs field, the members for this EVs research needed to young scientists like SNEV is very motivated to continue to work, the young scientists like you, like everyone, like Tom, Sarah. This group is amazing. Thanks so much to invitation. We have my support for everything, okay? Continues in science. It's difficult, but continues.
Thank you so much for your kind words.
Thank you so much. Thank you.
See you the next night, Ned.